Just introduce myself. My name is Jonathan Krieger. I'm the applications manager at BSI, and we've been doing this webinar series to give everyone an idea of PEAKS X Pro and what it can do. And so this one is going to be focusing on PEAKS Online and give you a little walkthrough of what PEAKS Online looks like. So what I hope is to show an overview of what types of analysis can be performed, the new features in X Pro, what is online, and I go through a demonstration and show some mixed species data that we have from published data sets and how that looks in online. So, because I can make this interactive, does anyone here use online? I don't know, you can just type in the chat, or if anyone's seen it, that's great, or if not, just let me know. But basically what we've done is we've really taken the new PEAKS algorithm and we've deployed it across two different methods. So one is the PEAKS Studio that everyone is most likely familiar with, which tends to work really well for smaller data sets or smaller files, but as these data sets get larger and larger, so does the computational power that needs to go with it to search it. And so where we've put a lot of work is to actually deploy this in Studio and in online. And then the online is really geared towards the high throughput, large file size type of experiments that can also be changed and edited on the fly. And I'll hopefully show you that. And so, as you can see, this crowd knows there are multiple approaches for identification of peptides and proteins in workflows, and they can be either spectral library searches where you can identify peptides and use a predefined library, so to say. One is to do a sequence database search, which you can match sequences from coding regions or whatever you're limited to is obviously what's in your database. And then you can do also Denovo Sequencing right off the Spectral, which is something that PEAKS does very well. And this will also allow you to identify things in non-coding regions like variants and splice sites, and as well as post-translational modifications go with everything. And so PEAKS Online is really designed to be a complete solution for proteomic analysis. And so this can include data-dependent acquisition derived data, as well as data-independent acquisition derived data. And so in terms of de novo sequencing, we can do this from DDA data in PEAKS Online, as well as it's a database search. And then for DIA, it primarily focuses in the library search algorithm, although we are developing a DDA based library search as well, just to make things even faster than they already are. We also have various quantification modules within PEAKS Online, just like PEAKS Studio, and this is label-free quantification, which you can do both DIA and DDA. Labeled quantitation, so this can include things like SILAC, TMT, iTrack. So these are primarily data-dependent acquired methodologies, but we are developing DIA SILAC quantification. And what's really where we spent a lot of effort is doing proper ion mobility integration for both, or I should say for all of the various vendors that provide ion mobility. So this includes HDMSE, timsTOF, DIA passive, and TIMS TOF runs, and FAMES data on thermo runs. And so one of the things that happens in the background, or you see it in Studio in the data refinement step, but it happens in the background of Peaks Online, is the feature detection in the software. And so the feature detection is really sort of the key in that this is where the starting point is of the identification and quantification process. And so what we do is we essentially look across the retention time M over Z and try to identify peaks or features by the isotopic distribution of carbon in a peptide. And so typically what we'll see is the M1, M2, M3. Sometimes you'll see the M4, the M5. But where we spent a lot of work is really the overlapping or improving of the feature detection for overlapping features. And so you can see in this image here, highlighted by the red, are 3 different features that all have portion of overlapping with each other. We've also now included this in the 4th dimension. So shown here is actually a timsTOF run. You can see at the top, we have the ion mobility dimension versus M over Z, as well as the retention time, and this produces our 4D feature. I'm actually going to skip to this slide. So what that really allows us to do in the identification is it allows us to understand that there are multiple features going on at any one point. And a lot of these features that overlap like this or have very close M over Zs are actually going to make it through the quadrupole, and together as one sort of species. And thus you're going to have a chimera scan with multiple species in those PSMs. And so what we can tell from the multiple features is that there are 2 distinct features here, and they're both in the same MS2 scan. And so what we can actually identify is the more abundant, or in this case, the higher intensity red feature, which actually sequences very nicely to the more abundant MS2 ions, and then the lower abundant MS2 feature, which also includes these peaks that weren't identified in the original. Matt, question, peaks works with MSE data. Does it work with water sonar data? I don't think we've ever tested it, Matt, because we don't have any of that data, but certainly if you have some and can provide it to us, we can try it or work on it, add it in as a feature. Perfect. Yeah, so you can certainly try it. I'll probably go into Studio before online, but we can certainly try it for you or work with you to get it installed. Okay, so what is Peaks Online? So Peaks Online is essentially a server-based search engine. And so what happens is your processing server is placed on either a high-performance computing cluster on your site or somewhere on a system like an AWS or Amazon Web Service or Google Web Service in the cloud. And essentially all the work happens there, and you direct all this through a Peaks client. And so the Peaks client is generally a web browser. We suggest Chrome. And basically what happens is your LC-MS MS data is ported into that client, or it's a repository is set within the client. You go into the client, you set up all your search parameters. The work happens in the background on the processing server, and then the data comes back to you via the client. And that's where the interactive results will set up, and that's what I will hopefully show you in the demonstration. And so what's the nice thing about POL? Well, there's a couple of things, but one of the really nice things is that it's linearly scalable. And so really that means that you can give it computing nodes as needed, and depending on your license. However, as you give the processing server more threads and more computing nodes, the performance increases in a linear manner. And so what I'm showing here is essentially 56 Orbi runs that were deeply fractionated into 672 180-minute MS runs, equating to around 29 million spectra, or 29 million MS. And so, you know, with a 32-thread version, it took about 10 days. That's cut in half with the 64, and in half again with the 128, and in half again with the 256. So, you know, the 64 to 256, that seems to be the range where most people are using online in, and certainly 28 million spectra in a day and a half or 2 days is pretty good. And so what's new in the PEAKS Pro versus the previous versions of PEAKS? So one of the main things, as I talked about this deployment that we have of Studio and Online, is that we've spent a lot of time ensuring that you have the same functionality in both deployments. And so this includes functions like Denovo, database search, library search, PEAKS PTM and PEAKS Spider, which a lot of people use, and as well as quantitation like TNT, SciLac, label-free quantitation. And so I will show you a slide that what we really optimize this for is consistent and good results across all the PEAKS platforms. Overall, in both versions, Studio and Online, we have new features, and this includes the iron mobility support for timsTOF, full iron mobility support for FAMES, and HDMSE. And Dan will be giving a webinar next week on that, specifically looking at 4D ion mobility in both Studio and Online. We also have improved the label-free quantification and match between run algorithm, which we call ID transfer. We've enhanced the spectral library search algorithm, specifically for general DIA data, but also ion mobility-based DIA data. And then in the Studio viewer, which you can download for free, we now have a spectral library viewer and editor. And so this can view and edit PEAKS generated libraries, as well as some third-party text-based spectral libraries. And we're always increasing this to add more support in there. And finally, advanced SciLac and label-free quantification. And so this comes down to really the improved feature detection, that we're able to do better MS1 quantitation on SciLac, and as well as label-free data. And so what I want to spend the majority of this time is actually showing you PEAKS Online. And so I'm going to share that window. So hopefully you can see my PEAKS Online. And so really what happens when you get PEAKS Online is you get set up, and you have a server address that you can access from a web browser. And then your administrator will, whoever you decide that to be, will set up a unique username for you. And the number of usernames that you get really depends on the license category that you get. But essentially, every user can get their own account. And so you can log in for yourself. You can save your password, obviously. And once you log in, you see your list of projects that you've been working on. And so what's really nice is it's quite quick, and you can work on the fly. But also, like Studio and other PEAKS products, everything is customizable. So at the top is sort of where you're going to have your main workflow. And so you can have, you have buttons to create new projects, to import archive projects, and then you have various settings. And so when you are done with a project, you can click on this one-click archive button, and it will be stored outside of PEAKS Online. And this really frees up space on your processing server. It's very simple to do, but if you ever want to bring that project back and reuse it, you just simply click on on this import archives and pull in your archive, and your project will be listed. I don't have anything archived here right now. And it will come back in just like it's an open ongoing project. So it's really nice. It's pretty quick to do, but it does allow you to clean things up sort of in the background. With Peaks Online, again, you have your option under the project window to create a new project, to look at your projects, or to look at shared projects. And so shared projects are projects that are shared amongst your team or your cluster of people. And so to share any project, you just simply click on this little person icon and it's instantly shared. And to revoke that access, you can unclick it. And so if you click shared project, these will be projects that are shared by other people in your group, but they are actually only editable by the owner. And then of course, you also have different settings. So most of the settings you can actually change on the fly, like adding in databases, enzymes, etc. But you can also do this ahead of time under the settings tab. And so to change a database, you can simply click on database, you can click the plus sign, and you can pull in any database you'd like. So it's just basically any FASTA file. You just click on the choose a database and pick your database file and hit go, and it will import it for you. If you look at the databases, this is our list of things that are already imported. If you want to pull that for whatever reason, it's very simple to just download it as a FASTA file, and then you'll get it. The other things you can do are generate spectral libraries, which I will show you how to do. Custom modifications. So just like in Peak Studio, we have the list of the Unimod modifications built in. So this includes common, uncommon, glycans, and artificial mods. But you can, of course, customize any modification you'd like or you commonly use just by clicking on the plus and putting in the modification rules. And so your quantification methods are here. The majority of them are all built in that you'll ever need, but of course, you can customize any quantification method as well as enzymes if you're using a combination or something new. And so it's really customizable to what you are doing and what your work entails. And so let's show a demo on starting a new project and the workflows and how it works and what it looks like. And so if you want to create a new project, you just simply create on the new project, give it a name and give it a description. And here you have two choices, one to create a new workflow or one to use an existing workflow. And so I'll show you about saving workflows, but what's really nice is if you're consistently running the same type of analysis with the same parameters and settings, you can essentially do a one-click and go, and that will save your workflow. So I'll show you how to do that, but essentially you create a new workflow, and then this is very similar to any other Peaks product that you know of, but essentially you can either upload your data from somewhere on your computer or wherever you're saving it, or you can select a repository. So I will show an example. I'll go here and I will pull in a file. So I'll pull in 3 files. So these files are timsTOF data, human yeast E. coli from the Jürgen Cox paper. And so essentially, because these are biological replicates of the same thing, but we'll want to put them in as individual samples. One of the nice things, so your options are to pull them in as one sample. So we would want to do this if these were offline fractions of some sort, which they're not, so we don't want to do that. We can pull them in as individual samples, which we do want to do. But here, unfortunately, this is great if you want to change the name and individually rename your files per sample. But the other thing we can now do in Peaks Online is take these files and click this button at the bottom. And essentially what we're telling it to do is to delineate based on file name. And so we're going to pull in all the file name because that's what we want to do. In this particular case, we're saying that there's a period. And so we don't care about the .d. And so when we upload that, the sample name now becomes the same as the file name. So that can save some time, especially for large projects. This is timsTOF data. So we will tell it it's timsTOF data and click next. And so on the next, you have your choice of whether this data is data dependent or data independent acquisition, or which workflow you'd like. Under the data dependent, you can do the de novo only. You can do the DB, which includes PTM and Spider, or you can include the peaks quantitation. And so essentially, let's do the quantitation, and you click next, and what you get is your standard parameters. And so these are going to be based on your instrument. So this is timsTOF data, so I will put in parameters for timsTOF data. You can select your enzyme. Your enzyme will really depend on anything you put here will override what we put on in this page, but these are all trypsin, so it doesn't matter. And then your digestion mode. Specific is essentially, I want every peptide to follow my enzyme rule. Semi-specific means I only care if one end follows the rule, and unspecific means both ends of the peptide can follow the rule. And you pick your database. And so you can either upload a database on the fly, as I said, you just click add a new database, or you can pick one you already have. And if you're using something like a UniProt or an sProt, the taxonomy files are built in already to Peaks Online, so you can just automatically pick what you'd like. So this data set is human, E. coli, and yeast. We'll pick those. And modifications are similar to any other, to Studio. You just simply add your modifications and define which ones are fixed and which ones are variable and hit confirm. The de novo will use the same parameters you've set unless you unclick this and reset different parameters. And what's really key to Peaks Online is you now have more control over FDR. And so at the PSA, you can control PSM or peptide FDR, your choice. I often prefer peptide, so I use peptide. And you can also control proteins by either minimal 10 log P score, or you can create a protein FDR score and say 1%. The other thing you have now is quantification. And so this is a label-free quantification study into a couple of groups. So it's actually 3 biological replicates and 3 biological replicates of different spiked in human yeast and E. coli proteins. And so this is where we will now group our biological replicates. We don't want to do it at the sample. We want to do it here at the label-free quant. So we'll put those into one group, and we'll put these into a new group. We'll change the color. So now we have blue and green. And you can rename this group if you want, A and B. And then from here, you have several options. So really, the match between runs is going to automatically enable an ID transfer if it's detected in one and not the other. And so you set your tolerance here, and I'd recommend using the same as your parent tolerance. And you no longer have to specify a retention time. The software will automatically detect any retention time shifts in the sample. So that's a nice new feature. And then at the bottom, you have several features that you can control what shows up on your display. And so at the peptide level, you can say, I want at least so many peptide IDs per group. So this would be, I want to see this peptide in at least 2 or 3 samples per group. And this is saying, I at least want to have X number of MS identifications per group. And so if I'm saying 2, that means that I have to have an MSMS identified from 2 of the 3 runs in each group. And so this can be really filtered up to you, and it's quite quick to change. And that's what I will show you as well. You can also choose this group coefficient of variation filter. And so this is basically going to pull out anything that is not as an outlier outside of the CV within the group. You can also filter things at the protein level. So you can remove outliers, and you can show things that are only significantly different between your groups based on an ANOVA test, if you'd like. And so you can change this significance factor. You can either use this 3 buttons and pick a value. You can go up or down, or you can type in any value you want, as well as fold change. And so when you're satisfied with this, if this is something you're going to use often, you can save this workflow. Let's call this Demo Thursday. And when you save that workflow, this will now, all these parameters will essentially be saved for you. So if we come back and pretend we're starting a new project, we can now hit select our workflow. We can pick the workflow we just created. Obviously, the files are not going to be saved because this is to do work on other files, but I will pull in the files. And when you hit next, everything is already here. The workflows we're already doing the quantification, and all the parameters, database, etc. Everything is saved. The only thing that is not going to be the same is your grouping, because we have new files potentially. If you do want to change something, you can either save the workflow as a new one, or you can update the current. And when you hit next, what happens? Oh, we do our groups. This group. So once everything's good, you hit submit. You can give it a name. Hit go, and essentially the system will start to process the data right away. And so first it'll load it, it'll do the refinement, Denovo, database, and LFQ. So what does the data look like when we're done? Oh, I'm sorry, not that one. When your data is done, you get your analysis with various nodes attached to them. You have your Denovo, your database, and if you did an LFQ, your LFQ. If you did a PEAKS, PTM, or a Spider, then they'd also be there. So looking at the LFQ node, actually, let's start with the database node. When you look at your database node, what you get is a summary chart of everything identified: PSM, peptides, sequences, proteins, etc. You get your FDR curve, and you get some statistics about the FDR, as well as mass error recalibration that occurred between the data. So this data is all off by about 8 ppm. We've corrected that. And we also have a chart of miscleavages allowed, as well as PTMs. You can, of course, look at any of the data. And what you get with PEAKS is sort of this overview summary. And so you can actually pick all 6 if you want. And so what you're saying is the protein coverage view for all samples together, and the intensity of red affects the number of spectral counts that you're seeing. And so the more intense samples are have a higher spectral count, and so on. If you wanted to look at individual samples, you can just at the top here click specified sample, sample 1, and you get your traditional coverage map. And of course, this is interactive. You can click on any peptide and view the spectrum of that peptide quite quickly if you go around, or you can actually double click on it. Oh, my double click is not working right now, but you can view the spectra, and you can actually flip through all the different precursors where this data was identified in the sample. So this basically peptide came up with two different features. Again, you can see a peptide table if you want. It takes a few seconds to load because it's the full peptide, but once you load it once, everything becomes cached within the browser, and so it comes quite quick after that. What's really important with this data set is actually the LFQ quant, because what we have in here is three technical replicates of two samples, and these two samples are essentially have different spiked in amounts of yeast and E. coli proteins. And so what you can see when you go to the label free is, again, your summary page, which tells you the number of quantified proteins, a volcano plot of those proteins, and then some information about the match between runs. So whether there's a retention time shift across your samples, so before and after alignment, as well as if there's an M/Z shift between your samples. And what you get is a graph showing the missing feature values. And so these are the missing features before ID transfer, and this is after. And so obviously with three technical replicates of a very ground truth, very well-established sample, we can get down to zeros or very, very low numbers. In a typical sort of perfect data set, if you will, these tend to be around 25 to 30%, and then these tend to be between 5 and 10%. So there is quite a big difference in the match between runs, and it does actually increase the identifications by quite a bit. When we look at the details, well, we can look at the proteins or the peptides of these proteins, and the volcano plot is interactive, so you can actually zoom in on a part if you'd like. And if you're interested in a particular protein, you can hover over and get what that is, or double click on it, and it will automatically navigate to that protein. You get your protein, you get your sample profile mini heat map, but if you hover over, you'll get the exact ratios, and then you get the group ratios, which includes however you grouped it, in this case, the 3 and 3. Any protein, again, you can also click on the peptide. You can see all the peptides that were identified for that protein and the ones that were used for quantitation. So we selected earlier a top 3 method, but you can change that. The only rule is that we will only use unique peptides to do the quantification on. And so if you want to look at the individual peptide, you can double click it from that view, and again, take a minute or two, and it will open the exact peptide as well as the extracted ion currents for all 6 of the samples. And so what you can see, you can either have the raw or the aligned XICs, but here you have the 3 lower samples, and here you have the 3 higher samples. And of course, you can go back and see your ion as well. And so what's nice, if we come back out, how does this data look? Well, if we go back to the protein level and we filter for yeast proteins, what you will find is they are all in the 1 human to 1 to 2 or 1/2 to 1 relation to the human proteins, which are in the middle, and they all form in this little plot here. If we look at the human proteins, which are all spiked in a 1-to-1 between the 2 samples, they're all centered around 1. And then if you look at the coli proteins, which are a 4-to-1 ratio between the 2 samples, they all sit up here in the 4-to-1 ratio. So it's nice. And you know, one of the really nice things about Peaks Online is how dynamic it is. So if you are here And let's say you want to modify those groupings. All you have to do is click on modify parameters, and you can come in here and change those groupings. Or let's just say you want to change the filter. So now we want to come here and we want to say, show me things that are 1.5-fold and statistically significant. And you hit save. Once you send that, essentially, what's nice is we're not redoing everything. All we're doing is that grouping and filtering. So that analysis is actually now done already. And you can click here and you can see we've regrouped. We're now only showing things that are significant. So that pretty much gets rid of all the human proteins for the most part. And we have our yeast, we have our E. coli proteins sitting here. And so the other nice thing about that is, let's say, oh, we've actually done 3 more runs. What do? Well, we can come back here, we can modify the parameters, we can come to data, and we can add in another file and bring it over. And the parameters will stay the same. And when you hit submit, again, what will happen is we will only do the refinement in the database and the searching on the new sample. And so that's a bit of a different departure from the studio, in which case everything gets redone from the beginning. Here, you would only do the new additions, which makes it a lot quicker. And so what's particularly nice for larger data sets is you don't have to wait for the data set to be finished before you run that. You can just run your sample right away without any issue. So now we've done this, what about DIA? Well, first thing we want to do with DIA is create a spectral library. So I actually have a sample set of timsTOF, yeast, E. coli, and human fractionated samples that I used to make a library, and that can be found here. And so this is basically an analysis where I have E. coli fractions in 2 replicates, human fractions in 2 replicates, and yeast fractions in 2 replicates. And we just did a simple database search. And so now what we do is we simply go to settings and spectral libraries and create a spectral library. Here's that search I just showed, and you pull it over and you give it a name and hit start. And it will automatically start to generate that library for you. If you'd like to view the progress, you can click on that and view. And depending on the library, that's how long it takes, but it's relatively quick for the most part to generate that library. So if you want to use that library, cancel our test project. We don't need it anymore. Let's cancel this library because it's not needed. And so what I have is the same data, but run in DIA mode, searched against the spectral library that I just showed you. I had just made it earlier. And so what you get when you do a spectral library search is a page very similar to your database page. With your FDR controls, your mass shift, you will see 2 additional figures. This is the regression. And so here you'll see a retention time regression for one particular sample as an example. And the red dots represent the internal retention time standard peptides that we have built into our internal libraries. As well, you'll see a box plot, and this shows basically the deviation from the found peptides to the index peptides throughout your run. And so you expect at the very ends, because everyone's ends of the gradients are largely different, to have bigger variation, but quite tight box plots through the middle. And again, just like a database search, you get the same view. You can pick a particular sample, you can look at any peptide, but this time what you'll see is your ion match. You'll see the extracted ion MS2 as well as the precursor. You have a little bar that you can see, you can change the correlation. So if you drop the correlation down, you'll see more MS2s, and this is how well XICs of the MS2 correlate to the precursor. And again, you can look at this for any fraction, any feature, or any sample that you want. So this was only identified in one. And sorry, I meant to show before closing it. If you click this, you can see the mirror image, which is the image between your library and your query, as well as a table of that info. And so all that's really put to use when you come to the label-free quantitation. And just like DDA, you get match between runs in the same manner. And if you look at your proteins, again, what we see are expected changes. So if we sort by the more abundant to the less abundant, you can click on that by peptides, and we can come here and show yeast. And these samples are actually flipped together, but there's our yeast majority of proteins, and we see the same thing with E. coli, which will be the opposite, 4-to-1. And so again, just like anything, you can go in and interrogate these proteins, and you can of course click on that and view the aligned MS2 extracted ion chromatogram from it. And so Peaks Online, it's great in that it's quick and you can add and subtract samples as you need. And where did my PowerPoint go? It's disappeared. But essentially, what's really nice is that it's really quite quick. And so really to add and remove files and to adjust FDR, both in just skipping over this, but to adjust FDR in both large projects and small projects, it's quite quick. To larger proteins, we've done studies with like these large CP-TAC data sets that have 500 TMT files, and we'll talk more about how the TMT works in the TMT mode. But what you can see, if I bring you back here and show you the CP-TAC colorectal study, is essentially 23 samples of TMT, and each sample is fractionated into 23 runs. And so I may have lost my connection to the office. I'm working from home, obviously. But essentially, having such a large study is no issue whatsoever for online, and you can keep pulling those files in. So I was hoping to show, there it is, okay. So momentarily connection loss, but essentially what we're seeing is 23 samples of 24 fractions of TMT, all normalized to one channel and grouped together into that one channel. And you can go through and look at the quant, but also, you can, where we've cut off everything here to really only show the things that are largely changing. But really the next step is to go through and group all the proteins. Right now they're grouped individually, but group them all by normal and tumor and et cetera. And obviously that data becomes a lot easier once you can find all the metadata from CP-TAC, which is sometimes difficult to find, but you can see, this protein is clearly changing in this group of samples and is just downregulated in this group of samples. And so, these large studies are kind of cool and interesting, and PEAKS Online really does perform a platform for people to do that with. And so with that, I'll end there given the time. But if you do have questions, you can always ask now or email support, and we'll be happy to get back to you. And this is the other webinar series that we have coming up.